the genetics lab for anatomy and physiology. So what we are going to be looking at today is gene expression and regulation. We're going to be looking at how DNA ends up making a gene, which is ultimately a protein. That's what a gene is. It's a protein. So how does DNA make a protein? And then we're going to discuss some things that can go wrong along the way. You're going to want to follow along with the notes packet to help you guide your information as we go through this video and the steps that are going to be involved. And then once the video is over, then you can click on the instructional video on the activity that you will be doing in order to reinforce the ideas that we just are about to discuss. So let's get started. First of all, a couple things that we need to discuss in terms of just reviewing basic DNA structure. So this should be reviewed to you. Remember that DNA is long chains. There's 2 strands to a DNA molecule. So on this diagram, you can see here's one strand and here's another strand, and then those strands are twisted into a helix shape. The 2 strands are held together through relationships based on nitrogen bases. There are 4 nitrogen bases, so guanine, adenine, cytosine, and thymine. And so guanine and cytosine are always going to hook up with each other. You can see that here, G's and C's. Adenines and thymines will always connect or bond with each other. So by looking at the nitrogen base, we can figure out what it's based on base pair rules, what the other strand would look like that allows those 2 strands to stay together in that double helix formation. There are individual units on the DNA strand, so the nitrogen base is part of the unit called a nucleotide, and the nucleotide is made up of obviously the nitrogen base, which can be either adenine, cytosine, thymine, or guanine, and also is made up of a sugar, a 5-carbon sugar called deoxyribose sugar. So that's DNA stands for deoxyribonucleic acid. And then there's also a phosphate group. So the sugar and the phosphate group make up this backbone. And then again, these nitrogen bases are going to be facing towards the center to allow it to bond with the nitrogen base on a different strand. So a couple different just basic vocab terms. So nitrogen bases, we just talked about those. So a series of 3 nitrogen bases will code for a chemical called an amino acid, and we'll talk about those in a second. Amino acids then, or chains of amino acids, are going to make up protein. A protein is essentially a gene. So when we talk about the gene for eye color, we're referring to the protein responsible for making that eye color. And then all of the genes connected together is what a DNA strand is. And we actually organize our DNA and our genes into sectional units, which we call chromosomes. So an amino acid is a compound that's coded from 3 nitrogen bases, a protein is a long chain of amino acid, and a gene is just a section of DNA that codes for an amino acid sequence of a protein. So a gene is just a section of that DNA that has a very specific function. Okay, so DNA is found in the nucleus of the cell, and if you remember back to your cell physiology activities, the nucleus is surrounded by a nuclear membrane. There's tiny little nuclear pores that allow some things to come in and out of the nucleus. But the problem that the cell has is that the DNA is too big to make it in and out of the nucleus. So it's pretty much stuck inside of the nucleus. The ribosomes, which are floating around the outside of the nucleus in the cytoplasm, those are actually where the proteins are made. So DNA is the instructions for making a protein, but the place where the proteins are actually made is outside of the nucleus. So the cell has to figure out a way to get the information from the nucleus out to the ribosomes, where the ribosomes can read the information and then put the proteins together. So it's kind of like having those instructions locked up in a safe, right? You would have to figure out how to get that safe open in order to translate those instructions, and that's what the cell needs to do. And it does that through a special molecule, which is called RNA. Sounds like DNA. There are some things that are very similar and some things that are different, but this RNA is really the go-between between the DNA stuck in the nucleus and the ribosomes, which is going to use those instructions to put a protein together. So RNA is kind of going to be able to go back and forth. It's going to be kind of like the security guard, right? He's got the key to the safe. He can open it and close it and get what he needs out. That's kind of how RNA functions. There are 3 types of RNA. We are going to talk about 2 of them today. The other one we don't really get too much into, but the first kind is going to be mRNA, which stands for messenger RNA. This is really, really important because this is what's going to connect the DNA, specifically the information on the DNA, and carry it to the ribosomes. Then we will talk about transfer RNAs, and these are going to be different types of RNA structures that are going to put the amino acids in the right order and in the right place. And then rRNA is ribosomal RNA. We scientists believe that ribosomal RNAs are used to help make the ribosomes, but there's still a lot of research going on exactly how rRNA works and what its function is. So we aren't really going to talk about rRNA today. We're going to focus on messenger RNA and transfer RNA. So RNA, like I said, it is similar to DNA. And then there are a couple important differences. So it is also made of nucleotides, so those units that are in a long chain. There's a sugar. The difference is that the sugar is a little bit different. So in a DNA, the sugar is a deoxyribose sugar, and in RNA, the sugar is a ribose. It still has the phosphate group, so that sugar and phosphate group kind of make up that backbone of the RNA. And then it also has nitrogen bases. The 3 major differences between DNA and RNA are that sugar is a little different. It's a different variety of sugar. RNA is single-stranded. It's not double-stranded like the DNA. And RNA will have a U or a uracil instead of a thymine. There's no thymine in DNA or in RNA. So there's still adenine, there's still guanine, and there's still cytosine, but instead of thymine, RNA has a uracil possibility. Now this is important because if I asked you, if I gave you a string of letters and said, is this a DNA or an RNA? All you would do is look to see, is there a T or is there a U? If there's a T for thymine, then it's a DNA. If there's a U for uracil, then you would know it was an RNA. And these base pair rules are also going to match up very similar to how they did on the 2 strands of DNA, except that RNA is going to match up with DNA, and A's and U's are going to join together, and C's and G's are going to join together. So here's how this process works. There are 2 basic processes involved in decoding the instructions on the DNA in order to put those instructions together to make a protein. So the first step is called transcription, and this is going to be the process of DNA becoming a messenger RNA, or a messenger RNA will be made from the DNA. And then the second stage is translation, which is going to involve reading the messenger RNA, coding or translating it into amino acids, and then making a chain of amino acids to form the protein. So we're going to go from DNA to RNA, specifically messenger RNA, and then we're going to go from messenger RNA to an amino acid, which then will link all of those amino acids together to form a protein. Here's how these processes are going to work. In your notes, you can see here that you should have this DNA strand that's right here. So what I've done is I'm giving you the nitrogen bases on this strand, one of the strands of DNA, okay? Basically, I've unzipped and I've unwound the DNA, and I'm just focusing on one of the strands in that double helix. Okay, everything that we're going to talk about now is also happening on the other strand of DNA, but if we diagrammed it showing both strands, it would get super confusing. So we're really just going to focus on what's happening at one strand. So just to orientate yourself here, you know, T stands for thymine, A is adenine, C is cytosine, so thymine, adenine, adenine, C, cytosine, guanine, cytosine. So you can see how these are representative of the nitrogen bases. So on the other strand of DNA, if we were to write it out, you know, it would be A, because T's and A's join together, T, G. You know, this one would be A, T, T. okay, and so on and so forth, but we're only focusing on this one. So what we're going to do is we're going to go over the process. I'm going to go over the process, in words first, and then I'm going to come back to that diagram in your notes packet and show you exactly what it looks like if we were to diagram this process out. So the first thing that's going to happen is transcription. So this is going to be going from a DNA to a messenger RNA. And so what happens is that a chemical or an enzyme, which is called RNA polymerase, is basically going to attach itself to the DNA, and it's going to be a very specific part of the DNA, which is called a promoter region. Not all of the DNA that we have in our bodies are used to make a protein. So we have sections of DNA that are not involved in the actual protein. So some of those sections are called promoters. So their job is to be a signal for the messenger RNA to join onto. We have some DNA which are kind of just like junk DNA. They kind of just fill in the gap. It's like filler. So not all of the DNA that we use or that we have is going to be used to make a gene, or is it an active part of a gene. We have other sections of DNA that aren't. So the RNA polymerase is going to attach to a region of the DNA that's called the promoter, and it's kind of like I mentioned, it's kind of like the "Hey, I'm here" button on a map. And then it's going to basically start traveling along the length of the DNA, making a messenger RNA alongside of it. So the messenger RNA will be made by using the base pair rules, and I'll show you how that works here in just a second. So A's will end up going into U's, because remember there's no T's on an RNA, and then G's and C's will connect together. And it will keep doing that until a stop signal is reached. At that point then, the messenger RNA actually ends up being released, where it can then leave the nucleus and go into the cytoplasm to find a ribosome in order to start taking those instructions and putting it together. So basically it's kind of decoding all of this instructions into its own sort of language, and then it's going to take that information out to the ribosomes. DNA and messenger RNA, they're all going to work in units of 3 nitrogen base pairs. So a codon refers to a group of 3 nitrogen bases on a messenger RNA. So we're going to go over to your sheet here with your DNA, and we're going to go over these 3 steps really quickly. All right, so here's the DNA strand that you have, right? So these are the nitrogen bases, and just remember that this is just one of the 2 strands of the DNA, okay? So the first thing that's going to happen here, and you can kind of put this up on top, and it's number 1, so it corresponds to the number 1 step on the PowerPoint slide then, is that an RNA polymerase is going to come and it's going to basically attach itself to that DNA, right? So it's going to come right into here. The next thing that's going to happen then is that the DNA is going to make a copy of, or it's going to make a messenger RNA. So this is step 2. So messenger RNA. And here's how it's going to do this. It's going to do base pair rules opposite. So a T is going to always bind with an A. So we're going to have an A here. An A is going to turn into a U. A C is going to turn into a G. So a T here, A will turn into a U, A will turn into a U, C's turn into G's, G's turn into C's, C's turn into G's, and so on and so forth, all the way down the messenger, all the way down the DNA. So it travels all the way down the DNA, making this a strand along the way. Now, if it's helpful for you, sometimes it's helpful just to write this out. So A will go to a U, a T will go to an A C will go to a G will end up going to a C, right? So if that's helpful, just to kind of help you write down what those rules are, those base pair rules. So at this point here, this is the messenger RNA. And so step 3 would be that this messenger RNA, that it's released from the DNA. So going back now to finishing up this step, transcription. The next thing that's going to happen is what's going to be called a modification put on the RNA, the messenger RNA. So two things that are going to happen is the first is going to be called a cap. So a cap is going to be placed. It's going to be at the very front of the messenger RNA strand. And basically this is a modified guanase, and it's going to help tell a ribosome where to attach. And then the second thing that's going to happen is a tail is going to be put on the very end of the messenger RNA. And basically a tail is a chain of anywhere from 150 to 200 adenines, so we call it a poly-A tail. So there's just a whole huge string of A's. And this is going to allow the messenger RNA to exit the nucleus and protects it from being disintegrated or damaged as it leaves the nucleus. So we're going to put this cap on, and we're going to put this tail on. The last thing that's going to happen then is going to be that we are going to get rid of any junk DNA, right? We don't want to have to go through the rest of this process decoding or translating the information if we don't need it, right? So if it's not important, we're going to take it out. So we put our sections of DNA into two categories. Introns are going to be sections of DNA that are not gene related, so they don't code for a protein. And exons are going to be those sections of DNA that do code for a protein or are an active gene. So basically we're going to remove, we call RNA splicing, we're going to remove all of the introns, and then we're kind of going to push all of the exons together just to make the rest of this process a little bit more efficient. And then at this point, the messenger RNA will be able to leave the nucleus. So again, going back to your diagram, we can kind of draw this in. So step 4 here is going to be the cap and the tail. So if we add this, put this cap right here, it's going to be right at the very front of that. And then we would add this tail right here. And remember that the tail would be 150, adenines all kind of in a row, if that helps you remember that. So anywhere from 150 to 200 adenines. And then the last thing that we're going to do, like I mentioned, is we're going to do the RNA splicing. So we're going to just assume here that this is a intron and that this one is an intron. Okay, so basically for step 5, we're going to do RNA splicing, which means we're going to take this part of the messenger RNA out, and we're going to take this part of the messenger RNA out. So we're removing this codon. So remember, a codon is a group of 3 letters on the messenger RNA, and we're removing this codon. So at the very end of step 5 of RNA splicing, our messenger RNA is going to look like this. It's going to have the cap. It's going to be AUG. We took that codon out up here, so we're going to just move on to GCG. We took that was an intron out, so we cut that out. CGU, CAC, and then UAG. Right? And then we have the tail that would be stuck here at the end. This is what our messenger RNA looks like at the end of transcription. Okay, so now that this messenger RNA can leave the nucleus, it's going to go out of the nucleus into the cytoplasm where it's going to interact with ribosomes. And the second type of RNA called transfer RNAs in order to code for amino acids and put the amino acids in the right place. Remember that a protein is just a chain of amino acids. Amino acids are going to be coded by codons on the messenger RNA, so those groups of 3 nitrogen bases. So there are basically 3 stages to translation. So number 1 is initiation, so all of the components are going to be brought together. This is going to be the ribosome, the transfer RNA, and the amino acids. Those are the 3 main components. The second is elongation, so amino acids will be put into place one at a time, and it's going to use the messenger RNA as a code to do that. It'll kind of lock it into place. And then termination is going to be when the transfer RNAs, the amino acids, and the messenger RNA all detach from each other in order to go their separate ways. So the transfer RNAs and the amino acids will be used in this process going forward, and but the chain of amino acids is actually going to be what the protein is. So these three general ideas. So again, we'll kind of go through this on this PowerPoint slide, and then we'll write it all out. So if we continue from where we left off, the mature messenger RNA is going to leave the nucleus and it's going to travel to the cytoplasm. And their ribosomes that are floating around in the cytoplasm are going to attach to the messenger RNA. And they're going to attach, so you can see this ribosome right here, it's going to attach using the second type of RNA, which is called transfer RNA. So transfer RNAs are kind of these little sort of square-like structures here, and you can see that they have this little circle attached to them. These circles are actually the amino acids. Okay, so amino acids are going to be carried by these transfer RNAs. And then the transfer RNA also has a group of 3 nitrogen bases on the bottom, and this is what's going to, these are called anticodons because they're found on the transfer RNA. These are what is going to attach to the messenger RNA in order to put each of these amino acids in the right order. Okay, so here's, you can see the transfer RNA has the specific amino acid attached to it. Once we get all of the amino acids lined up in the order that they should be, there will be a code that tells it to this process to stop. And at that point, then everything gets released and the amino acid now becomes a protein because all of them are in the right spot and they're locked into place. And the protein can then go do whatever job it is that it's supposed to do inside of the body. Proteins that are made in this way, once they are kind of released, they will change their shape to become very specific. Proteins have very specific shapes. So this kind of final piece is just like putting the finishing touches on that protein. And that's done on that endoplasmic reticulum. All right, so here's how this ends up working. So what I'm going to do is I'm going to just rewrite this messenger RNA down here, just because it'll make it a little bit easier to kind of see what's happening if we make it a little bit bigger. Okay, so this is the messenger RNA. And I just copied it, right? So we are now out in the cytoplasm. So what happens then, like I mentioned, we have this ribosome that's kind of going to sit on here. So we can put ribosome on here. So ribosome is where proteins are made. And then we're going to have these structures called transfer RNAs, right? So they kind of look like this. And then they have this circle on here, which is an amino acid, right? So this is a transfer RNA, and then it's got attached to it an amino acid. And then down below, it's also going to have 3 nitrogen bases that will pair up with these ones. So all of these transfer RNAs, I'm just gonna draw all of these in here quickly. All of these transfer RNAs are floating all over the cell. Okay, so this transfer RNA is going to have, if you look down at the bottom, we're going to go opposite again. So G, this one's going to have a C, this one's going to be a G, and this one's going to be a C. So that's how the transfer RNA knows where it's supposed to go on the messenger RNA strand, because it's going to look to find its pair. Okay, so this one is going to be a G, this is a C, this is an A. This is going to be a G, this is going to be a U and a G, and this one's going to be an A, a U, and a C. And then this one, which we forgot in the beginning, is AUC. Okay, so these are the anticodons that are used to kind of attach itself. It finds, so this AUC is floating all over the cell, and it's going to like move along the messenger RNA until it finds a UAG, because that's its pair, and it's kind of going to lock itself into place. So, you can kind of draw a line here showing that it's going to bond together, right? So these ones will bond here. This one's going to bond here. So you can kind of see how that works. So the 3 on the transfer RNA is the anticodon. The group of 3 on the messenger RNA is the codon. Okay, so now we got to get to this amino acid. So this is kind of step 8 was this transfer. So now we're going to go to how do we talked about how these code for an amino acid. So this was step 9. How do we know what amino acid goes into place? There are 22 types of amino acids. So how do we know which amino acid is going to go into which of these transfer RNAs, or which transfer RNA is going to carry what amino acids? Well, we're going to use this handy dandy chart that I will show you here in a second, and we're going to use these codons to tell us what the amino acid is. So I will show you how this works. Here's an amino acid chart. Along the side is the first of the nitrogen base, along the top is the second, and along the back here, or along the side, is the third. So our first messenger RNA, our first codon, was AUG. So what we're going to do is we're going to look for the A under the first base here. So here's where the A was. Then we're going to find the second nitrogen base, which was a U, over along the top. So here's the U, so we know we're going to be in this entire box here, right? And then we look for the third one on this end, and we go over, and we know that AUG stands for the amino acids methionine. Okay? So the next amino acid, or the next codon, was GCG. Right? So we're going to use our chart. So find the first base along this side. So here's the G. Then we have a C. So we got to look along the top and come down. Right? So come down here. And then G is on this side. So we're going to go over. And here we are. So GCG equals alanine. And we're going to continue along until we come to the end. So what I want you to do is go back to your sheet and kind of fill in the rest of the nitrogen bases based on what you had. So this was the second one. The third one was GCU. The next one was CAC. And the last one was UAG. So try and find the amino acids for each of these using this chart. So here was the methionine, here was the alanine, here's a cysteine, and usually we just kind of put the first 3 letters because some of them have big words. This was the histidine, and this one coded for the word stop. So basically what this stop is that it's telling the process, it's telling the cell that this gene is complete, and the next series of 3 codons, that is actually the start of another gene, because all of our genes are in this huge long roll on our DNA, right? So maybe the gene for our eye color is right next to the gene for our hair color, and maybe that's right next to the gene that determines how much protein we make, that determines our skin color. So like if we're using that example, then once we get to a stop, we know that this gene and this protein, because protein and a gene are the same thing, is done. It's been completed. And so the finished protein, so what happens at this point then is that all of the messenger RNA, or all of the transfer RNAs get released, all of the messenger RNA gets released, and what we have left is a chain of these chemicals, which are amino acids. It's a methionine, an alanine, a cysteine, a histidine, and a stop codon. This is the finished protein. These amino acids in this specific order. And now this protein will go, it'll change shape, it will become very specific in the rough endoplasmic reticulum, and it will go into the cell and into the body to do whatever job it is to do. So we started with a DNA strand, which was instructions on how to make this protein. This particular section of DNA is a gene. It's a section of DNA. A gene is a section of DNA that codes for a protein. This DNA is stuck in the nucleus, so we had to get it out, and in order to do that, we had to make a messenger RNA strand, and we made that strand using base pair rules, going opposite, remembering that there are no T's in RNA, only U's. U's replace the T's. We did some modifications to that messenger RNA in order to help it get out of the nucleus. We added a cap, we added a tail, we did some RNA splicing where we got rid of junk that we didn't need. Then the messenger RNA left the nucleus to go into the cytoplasm where ribosomes that were carrying transfer RNAs came in, attached themselves to the messenger RNA, and those transfer RNAs carried amino acids. And then we release everything, leaving this chain of amino acids, which is the actual finished protein. Some things that students get confused about, when you are coding for an amino acid, you always want to use the messenger RNA. Okay, when in doubt, use messenger RNA. So sometimes students look and they think that they're using these letters, so AUC instead of UAG, but really these are just to kind of hold their place markers. They kind of hold it in place. Think of it kind of like Velcro. It's just Velcroing this codon or this amino acid into the right order until it's all done and everything can just get slammed together. So always use the messenger RNA when you're using that chart to figure out what the amino acids are. Messenger RNA, messenger RNA, messenger RNA. Those are the big ones. So the next question is, what can go wrong with this process? When things go wrong, we call these mutations, and there are different kinds of mutations. But basically, a mutation is going to be a problem that happens that will originate in the DNA, but because it originates in the DNA, then the messenger RNA that is made is not right, and then the amino acid that the messenger RNA codes for is not right, and if the amino acid isn't correct and if it's not in the right order, then the protein isn't made properly or correctly, and then sometimes the protein then can't function, and that's what leads to disorder or disease. So mutations are changes in the DNA. It's really important to understand that all mutations start in the DNA, okay? Not in the RNA, it starts in the DNA. So there are two kinds of mutations that can occur, broad categories. One involves the nitrogen bases, so that's going to be the A's, the T's, the C's, the G's. And then other mutations are called chromosomal mutations, which has to do with the process of meiosis, and we're going to talk about that in the next lab. But the types of mutations that can occur that involve nitrogen bases are two, substitution or frame shift. So substitution mutation is going to be when one nitrogen base is replaced with another. So on your DNA sample, you started out with TAC, was the first group of 3, while maybe the DNA in a substitution, it's actually CAC, and the T has been replaced with a C, or it's been substituted. The second kind is called a frame shift mutation, and this is going to be when nitrogen bases are actually added into or deleted from the DNA. Now, substitution mutations are typically not nearly as serious or devastating because sometimes the substitution, like if you go back and look at that chart, you can still make histidine using a lot of different combinations. So you might have a substitution and it still might code for histidine and it might be fine. And substitution mutations only involve one protein, right? If one nitrogen base is changed, it's only going to involve that protein that's being made. Once you hit that stop codon, then whatever occurs after that should be fine. Frame shift mutations are devastating, and that's because if you add a nitrogen base or you delete a nitrogen base in a DNA, everything past that point now, all of those groups of 3s have been shifted. So everything past that, every single protein on that DNA strand, every single gene is not going to function the way that it should. So frame shift mutations are really devastating. Mutations can occur in both types of cells. So they can occur in your germline cells. So these are going to be in the sperm or egg cells, and typically these will be inherited from your parents. And then they can also be found in somatic cells. These are the non-sex cells in your body. And these are going to not be passed on to your children. So for instance, skin cancer or melanoma, that would be a mutation that occurs in our skin cells, but because it's not in our egg or our testes, or I'm sorry, in our ovaries or our sperm, we will not pass melanoma on to our children if we have it. However, those diseases that are passed on or are hereditary are found in our egg or sperms. Those will be inherited from parents. Like I said, the effects of mutations, you can have a neural or a silent mutation. It means that it's found either in an intron or it's a mutation that occurs, a substitution, but it doesn't actually change the amino acid. The proper amino acid is still coded for and it's still in the right order. So these are called silent mutations. We have tons of silent mutations in all of our DNAs. We get silent mutations just from going outside, quite honestly, but a beneficial mutation results in a protein that improves structure or function. So you might have a mutation that actually becomes helpful. And then harmful mutations are going to be proteins that are not made completely or are incapable of functioning because they don't have the proper amino acids in the proper places. Two types of chemicals that can cause mutations. A mutagen is a physical or a chemical agent that can cause damage to DNA. So it can lead to mutations. That's what a mutagen is, but doesn't necessarily lead to the formation of cancer. So you can have a mutagen that will just cause a disorder or disease that's not cancer-based. A carcinogen is a physical or chemical agent that causes problems with the cell cycle that then leads to the formation of cancer. So mutagens damage DNA. Carcinogens are going to mess with the cell cycle. So there'll be cancer-specific types of situations. So the next thing that you're going to want to do then is just watch the quick video. I know this was a long one, I'm sorry, about what you need to do for your actual lab activity. If you have questions about this process, please feel free to ask.